and I'm with Vincent. And today we're going to show you how a genetically inherited trait will affect the population under the Hardy-Weinberg principle, and how to calculate the allele frequency using the Hardy-Weinberg principle. We will then compare our results to the North American average and show you how allele frequencies change over 5 generations within scenarios of natural selection, heterozygous advantage, and genetic drift. So the materials you're going to need for this lab are PTC paper, control paper, a coin, and note cards. So, step 1. You're going to take a piece of PTC paper, which is this right here, and you're going to put it on your tongue. So, what you're trying to see is whether or not you can taste the bitterness, and I actually can't. So, you're going to record the results into your first table. And now the second step is to take a piece of this control paper and put that in your mouth as well. And what you're doing here is comparing the taste from the two papers. So if they taste around the same, then you can't taste it. But if you taste like a really bitter, I guess, taste, then you have the gene for it. So you're going to record the results into your table 1 to the analysis section. And you're going to get the results for your entire class, and you're going to record that into table 2. Now you're going to tally the results for the entire class and calculate the frequencies for each allele using the Hardy-Weinberg equation. Now you're going to do that in the analysis section. And be sure to show your work. Okay, now part 2 of the experiment is testing the Hardy-Weinberg principle. So case A is testing the ideal population. So you're going to have 4 index cards, and the reason why we have 8 here is because 4 is for you and 4 is for your partner. And now you're going to take 4 of them, which 2 are labeled uppercase A and 2 are labeled undercase A, and you're going to shuffle them. And then you're going to flip them over so you can't see which card you have. And you're going to do that, you and your partner are going to do that. So once you do that and you flip them over, you're going to draw the top card. And for this case we have heterozygous. So whatever you have, your next generation is going to mirror that. So since we have a heterozygous, our genotypical ratios stay the same. So now you do the same thing. You record it into your table, and then you shuffle again and you redraw. So then you redraw it again, and you get the same thing. And just for the sake of time, I'm only going to do this twice instead of 5 times. Alright, so case B is selection. And so the last exercise was conducted with ideal parameters. So this is case B, selection. Now this is a more realistic situation than the one we did before. So if you select a homozygous recessive individual, then it automatically dies. And if that's the case, then you have to draw again. And if all the lowercase a's are eliminated, then you have more over here. So you do the same thing as before. Yeah, you shuffle it. You shuffle each stack. Okay, so now you're going to draw, and since this is homozygous recessive, then they die. This individual dies. So then you automatically redraw, so that you get heterozygous. So you have to have the same genotypical ratio as before. So you take, you have to make sure they have the same, 2 big A's and 2 small A's. You have small A, and you have another small A. So you do the same thing 5 times. You just shuffle, and for each recessive individual, it dies. And to keep the population size, you have to have 2 surviving offspring. So you can't have all homozygous recessive. Okay, so you do that 5 times, and then you record it into your table. Now, case C is heterozygous advantage. So you do the same thing as before. You eliminate the homozygous recessive, but now every time you draw a homozygous dominant, you flip a coin, and if it lands on heads, then that individual dies. And you do the same thing as before. You, if you get like homozygous dominant and it dies, and you have to redraw. So let's demonstrate that here. So now you draw it, and we got homozygous dominant, which means that we have to flip the coin, and heads means that it dies. So it got tails, which means that it lives. So you write that down into your table, and you do the same thing 5 times. And it's the same as before. You have to have 2 surviving offspring. Okay, that's it. Now, part D of this experiment is genetic drift. And genetic drift is a phenomenon in which an allele is lost solely from chance instead of through selection. Now, the most important factor in genetic drift is population size, because smaller populations will obviously have a much greater potential for genetic drifts. Now, we can't actually do this section because we don't have a class to compare the populations with, but we're going to give you the instructions anyway. So you have to have, so your instructor will divide your class into several smaller populations. In your population, you'll follow the mating procedure with the first exercise, and you do that for 5 times, for 5 generations. And for every genotype you record into your table. And now the last step is to combine your group's 5th generation result with other small populations and calculate a new allele frequency. So that will show you the genetic drift.
Alright, so this is our data for the first part, where we tested the tasting of the PTC and the control paper. Now for this part, since we didn't have a whole classroom to compare our results to, we just used the Brandon's family, which consists of him, his mother, and his brother, and let's just include me for the sake of this experiment because I tasted it too. Now as you can see here, for the tasters, only Miss Lorena, Brandon's mother, and I could taste it, and the 2 people who couldn't taste it were Brandon and Andrew. So that means we had 50% of the population who could taste and 50% that couldn't taste. Now, we were able to determine the allele frequencies of this. P for dominant, which means you can taste it, and Q is for recessive, which means you can't taste it. Now, this trait for being able to taste the bitterness in the PTC is a dominant trait. Now, here I want to show you how we use the Hardy-Weinberg equation to solve for the frequencies for this. Now, this is my calculations for it. Now, just wait for the camera to zoom. Right, now, the Hardy-Weinberg equation is as follows. P squared plus 2pq plus q squared equals 1. Now, this is for populations that have no genetic drift or changes in population. The gene pool is constant. There's no migration, net or in. There's no net mutation either. And this is used for theoretical purposes to study the effects of mutations or whatnot on a population. Now, p squared is your dominant, 2pq is your heterozygous, and q squared is your recessive. Now, what you want to do for this equation is you find q squared, which is the percentage that we found for the recessive, which was 0.5 or 50%. Now this is for recessive, remember. Now, to solve for the allele frequency of the recessive, you need to take the square root of it. So the square root of 0.5 is 0.707, and that's the recessive allele frequency. Now, to find the dominant allele frequency, we use this other equation, p plus q equals 1. And so we know what q is, we plug it in, and to solve for p, just use simple algebra. You should be able to do this if you're taking AP Biology. You just do 1 minus 0.707, you get 0.293. And now we have our dominant, which is 0.293, that's the dominant allele frequency. The recessive allele frequency is 0.707, and the heterozygous, which is just multiply 2 times p times q, and we get 0.414. And all of this added together is equal to 1. Now, for the other parts of the experiment, we did the same. Brandon and I, we ran the experiment by ourselves, and this is the results for part 2, the case A, and we determined the allele frequencies and whatnot. We also determined using the Hardy-Weinberg equation, but we decided not to show it here because that would be excessive, and since I already showed it how to do it in the first part, you should be able to use the same equation to solve for the allele frequencies for each case. Like case A, this is case B with selection against the homozygous recessive, and this is case. Alright, I'm sorry. This is also case B, case C. This is for heterozygous advantage, and the next one is case D for genetic drift, which we weren't able to perform accurately because we didn't have a whole classroom. Now, in your cases, you guys would fill out the class frequencies and the final class frequencies, and you would compare it with the rest of the class. Now, for the first part of the experiment with the PTC, we're supposed to compare it with the American average, North American average. And the North American average for people who could taste PTC is 26.5% of people cannot taste PTC. That means 26.5% of people have the recessive trait, and they express it because they're homozygous recessive. And this doesn't match our data because our data pool was very, very small compared to the data collected in the North American census. And that is the main reason why our readings were not accurate with what the North American averages were. Alright, thank you for tuning in and listening to our experiment and watching us perform it. I hope this helps you out. And